मैंने माइक ऑन नहीं किया. So this is the third and the final live session for the course Enzyme Science and Technology. So you can post your queries. You know that the exam is very close. So it is a good idea to clarify your doubts so that you will be in a better situation to answer the questions. And in case there's any aspect which is not been covered or which is not been clarified, uh, just by the lecture also, we can actually discuss that and we can clarify the doubts. I have few of the questions from the students, so that also I'm going to take up. But I want that everybody should join and then we can actually address the questions so that everybody will get benefited. So we have the Ishan Banerjee. And Ishan, I think I have seen your question. So I'm just waiting for some more people to join. Otherwise, within a minute, I think we'll discuss your question.
Okay, thank you, sir.
Yep. Ishan, can you turn on your camera and introduce yourself? Let me share my screen and so that we can discuss the question from the Ishan. Okay. So Ishan, can you repeat your question again?
Sir, my question was regarding just 1 second, sir. So I will open the page, I think, in my computer. 1 second.
What you have said, I will read out. Okay. You said that you don't understand how we ensure that a gene in the transformed bacteria is under the control of the lac operon, so that an inducer like IPTG can overexpress the gene in E. coli expression system. Which criteria of a good vector is essential for this? So it actually has two questions, right? One is, what is the good criteria for selecting a good vector? And the second is how you know that the gene of your interest is under the lac operon, right?
Sir, the criterion part you have discussed, sir, in the like in the lecture, but like the main question was like, in which of these criterions was like the most important for that part?
Okay. So first of all, I think let's first discuss the lac operon question. Okay. So lac operon question. फुल स्क्रीन मेरे में तो है. Make them. Well, okay. So sorry about this technical problem. Okay, Vishal. So what your first question is that how we know that the gene is under the control of lac operon, right? So
Yes, sir.
so for that, see, first of all, you have cloned the gene under a particular promoter, right? And that promoter has the component of the lac operon. So in that case, there's no doubt that your gene is under the control of some lac operon or something, right? The under the natural conditions, for example, if you want to know whether the gene is under the control of lac operon or any other operon, right? In those cases, things are a little difficult. Things are more different than if you are trying to know the same question from the vector, because vector anyway you're making the vector in such a way that you know what kind of promoter and operators are operational in that particular vector. So when you want to understand within the organisms, then what you can do is you have to see, for example, you have right, and imagine that this is a bacterial chromosome, and we know that this is the gene, right? And this gene is suppose we have cloned the actin gene, right? So this clone actin gene is the bacterial actin. Okay, so I want to know whether the bacterial actin is under the control of lac operon or not. Or indirectly, I'm saying that whether the lac operon is controlling the expression of bacterial actin or not. Okay, So we have a couple of options. Okay, one is, sorry. So we what we can do is you can take this bacterial culture, right, and you can add, so you can grow them into minimal media, okay, so that you can be able to control the inducers. And what you can do is you can grow them in the minimal media without carbon source. Okay, so you keep the carbon source, but once the bacteria is grown, then you can just remove the carbon source. And now what you can do is you can just add the carbon source as per your choice. So we have a couple of options. Okay, so what you can do is you can add the carbon source like lactose. Okay, so when you add the lactose, and if it is under the action of lacoperon, what you are, and then what you can do is you can actually be able to analyze the expression of bacterial activity. Okay, so there are a couple of options through which you can actually be able to see the expression. You can see the expression at the mRNA level. So you can actually do the northern blotting, and that will give you the idea whether the RNA is forming or not, right? If you want to be interested in early events of expression, if you want to do the Western blotting with ideally what you're going to see is this. You're going to see this is the minus, this is plus. So this is the place where you have added the lactose, right? So what you're going to see is that there will be no, so there will be all the bands, right? And in this case, there will be all the bands except there will be a band at 45 degree or 45 kDa. Okay, so that will say that there is a induction of this particular protein, which is under the control of lacoperon. Now, every experiment when you are going to do is you are actually going to use a control experiment, right? So we have the negative control, right? So we have negative control where we have not added the lactose. Then we are going to have the further controls, right? So that you can actually be able to verify. So this is the positive control, that you are actually adding the lactose, bacteria is utilizing the lactose, and that's how it is giving you the protein of your interest. The second is that you add the lactose, right? And along with the lactose, what you can do is you add very high quantity of glucose. Now, what will happen when you have the lactose and the glucose combinations? The glucose is preferred substrate for glycolysis. Okay, so the glucose is preferred substrate for glycolysis and even downstream Krebs cycle and all other things, right? Because glucose is easy to metabolize compared to the lactose. So what bacteria is going to do is going to utilize glucose instead of lactose. So in that case, what will happen is that, and if you again analyze the DNA, right, the protein sequences, right, what will happen is that you are actually going to get this, because you are now what you're doing is you are suppressing the activity of the lacoperon. So you are still having the lactose into the system, but it is not going to work because you have the better substrate for the enzyme. So the inducers and all other components of the lacoperon is not going to be present, and that's why it will start using the glucose. So that will be a verification experiment to say that it is basically the lacoperon only which is responsible for the production of this particular protein, not any other operon. Okay, because if it is under the expression of any other operon, if it is not under the lac operon, right, then you will still be able to see this band. Okay, otherwise what you're going to see is you can add like different amount of glucose. So what will happen is initially you will see this much, then you will see this much, then you will see this much, then you will see this much, and ultimately it will get disappear because the bacterial machinery is more producing the enzymes which are related to glycolysis and Krebs cycle rather than the enzymes which are responsible for the lactose metabolism. And this is some kind of verification what you can actually be able to do to understand whether the gene is under the control of lac operon or not. The second question is more about how you can be able to choose. Are you convinced with this?
Sir, I have understood very well, sir.
Okay, so this is just an example for lac operon. You can actually explore the similar questions even for other operons also, and even for other inducers also. The only criteria is that you should have an inducer and then you also should have a repressor, so that you can actually be used these combinations to design an experiment of similar kind. Now the second question is what criteria I should use to select the plasmid, the vector actually, right? So I think I have discussed in detail, but
yes, sir,
for vector, yeah, vector, you have two major criteria. One, the size. The second, the expression. Because if you're talking about the expression vector, right? These are the only two desirable criteria, and definitely you need a selection pressure. Okay, so selection marker, because without selection marker anyway you will not have any vector. So selection marker is present everywhere. So if you have the choices of vector from, like suppose you have 10 vectors, selection pressure will always be there. Okay, so you can actually be able to choose the size and what is the level of expression it is going to present, and what kind of inducer you are using. Okay, because inducer is actually not going to decide, inducer is going to decide the cost of production, right? Suppose you are working with the IPTG based inducers like T7 inducers, T7 promoters, right? Then the cost is going to be more because the IPTG is very costly. But if you are working with for example, if you are working with vector which are T7 promoter, right? The cost is more. If you are working with the T5 promoters, the cost is less because T5 promoters are not inducible promoters. They are actually going to give you the protein as in terms of growth. So you don't need any inducer. You just grow the bacteria and it will actually going to give you the proteins. Size is also very important because size is actually going to decide how good or how easy it is to transform the bacteria or transform the host. And that is very important because if you are actually working with a vector which is like suppose you are working with a vector which is like 8 kb, right? Then it is very difficult to transform any vector or any recombinant DNA which is like more than 10 kb. Okay, so if you are working with a vector which is 8 kb, then you only have an option of adding the 2 kb max, because if you add more, then the transformation efficiency will go down to such a drastic level that you will not get the recombinant DNA, you will not get the recombinant host cells, and if you don't get the recombinant host cells, the whole experiment is actually not going to work out, right? Because then you will not get the positive clones and other things. So this is just an idea. So if you are doing it in the lab, then size and the inducer, or I will say how it is actually giving you the protein. So inducer is very important because in case some places where you don't have the, if you are working in a lab where you don't have money, then it's better to go with the T5 promoter. The other advantage with the T5 promoter is that you don't need a specialized host, any E. coli will work because you have the T5 promoters present in every E. coli. Whereas with the T7 promoter, you require the specialized host, like you require the BL21 D3, right? So if you require the specialized vector, these vectors are also, these hosts also you need to require. Okay. So is it clear now?
Yes, sir. I have understood, sir.
Okay. Do you have any other question related to course or related to exam?
Yes, sir, I have a question, sir. Sir, the primers for each reaction are ordered online. So does this have to be done for each time a new PCR for a new gene is run? Because we have to like make a new primer every time we have to run a PCR.
Mm-hmm.
So like these like primers, every time we run a new PCR, we design a new primer forward and reverse. So all of these have to be ordered online. Is it like so?
If you are going to do every time the new gene or new fragment of the DNA, then definitely you have to order all the primers online every time.
Okay, thank you, sir.
Yeah. Okay. Okay, Isha.
Another just one last question, sir.
Mm-hmm.
Sir, during forward and reverse primer generation in the computer, I mean, when we do it in words or like in the computer, we add restriction enzymes to the primers, sir. Does
Mm-hmm.
the same restriction enzymes need to be used for cutting the DNA and vector for in ligation to work in like the practical lab too, sir?
Okay, can you repeat? Because the voice is a very difficult. It's a difficult thing actually getting here. So because the speaker is not good actually in our facility.
Okay.
Can you repeat again?
Yes, sir. So during forward and reverse primer generation, we need to add restriction enzymes to the primer, sir.
Mm-hmm.
Does the same need to be done? Does that mean that we have to use the same restriction enzyme for the cutting of the DNA and vector for ligation too, sir?
Yeah,
That
definitely.
one which you
Because
added.
otherwise, how you will be able to clone if you don't have the same restriction enzyme?
Okay, sir. That was like I think like so. I mean, yes, of course, like restriction enzymes have their own specificity, sir. But like this, I was confused about how like they change
So
text data.
if you want to integrate any DNA into another DNA, then they should have the sequence complementarity, right? And sequence complementarity will come only from the restriction enzyme, because one restriction enzyme will have 5' overhang, the other restriction enzyme will have the other DNA will have the 3' overhang, and that's why they were actually going to come together and give you the recombinant DNA. For example, if you have EcoR1 in primer and the vector is having the hint3, they will not have the complementarity, right? Because
No, sir. Yes, sir.
those fragments will not match each other.
Yes, sir. But for DNA and vector, it will be same, right? So, but for the primer, the one which we added in the primer in the text file, that too has to be used in the experiment, right, sir?
Mm-hmm.
Okay. So that was like I understood, sir.
Yeah. Okay. Fine. Okay. So this is all about from Ishan, right? And Ishan, what you're doing?
Doing bachelor's in science, sir.
BSc.
Yes, sir.
From where?
Biburua College, Guwahati, sir.
Okay. Okay. Good. So all these are you are going to use in your experiments?
Sir, so for my dissertation, I actually did an enzymatic study on this, our toxicity test. For that, I had to do a enzymatic study. But for sir, mainly I'm doing so that I can learn more about like enzyme and like the biotechnological processes for better, sir. I can do better in the
So
competitive exams.
what's your plan for after BSc?
So after BSc, sir, I'm thinking I will sit for the competitive exams and I will do a master's and things like that, sir. I will give the competitive exam.
So you want to do MTech?
I want to do a master's in science, sir. MSc or like I will give try.
MSc. Okay.
Yes, sir.
Good. Okay. So, this is all about in today's live session. I think we don't have any more questions from the other candidates, and I will wait for another a minute or so in case somebody show up and ask the questions. Otherwise, we'll conclude this session. And Ishan, you are fully aware of what kind of format is going to come for the Enzyme exam. Are you sitting in the Enzyme exam?
Yes, sir. I'm taking the exam.
Okay, so just my personal advice would be that you should go through with the whole content rather than specific contents. Okay, that this content looks more important, the question will come from this content and all that. You are going to see the questions from starting from the module 1 to module 12. You are not going to see any kind of focusing on any part or something, because every part is favorite for me actually. So we are going to see at least 10 questions from module 1, 10 questions from module 2, like that. So you're going to see 100 questions approximately.
Sir, just one question, sir. Kind of like out of, I think, syllabus of this course, but like I've been like trying to understand this, sir. But sir, how are so signaling pathways in like organism? So the way of studying them is the signaling pathways which happen inside, and so actually to like make a drug or an inhibitor of an enzyme, we have to like some idea about the signaling pathway of what happens in our organism, right, sir? So like this molecule is getting converted into this molecule which is assisted by enzyme A, B, and C. So how are like, can you like in a very brief way say that how are like these pathways actually studied?
So what you want to know that how people know that, okay, if I am adding the insulin to the beta cell, what are the different proteins are getting phosphorylated, and how they know that which one is first and which one is second, right?
Yes, sir.
How we know that this is actually the insulin signaling pathway, right? Where you have the receptor on the cell surface, then you have the SAP kinases and SIC kinases, and then you have the all the other kinds of MAP kinases and so on, right?
Yes, sir. Yes, sir.
That I think I'll share my screen, and we'll discuss, because it's an interesting thing actually, but it's not that straightforward that you understand. So let me share, and we'll discuss.
Thank you, sir. This was kind of out of like syllabus what you're discussing. Thank you, sir.
No, it's okay. It's okay. Okay, so imagine the same example of insulin. So insulin binds to the insulin receptor. Insulin receptor is a tyrosine kinase receptor, right? I'm sure you all did know, right? Tyrosine kinase receptor. And this is a monomeric receptor, right? So first question comes, if you add the insulin, where it is binding, right? Because it is going to bind either to the cell surface receptor. So imagine that this is a cell, right? So we have two choices. One, insulin can bind to the cell surface receptor, or insulin can go inside and bind some intracellular receptors, right? So what we're going to do is, you are going to take the insulin and we are going to label the insulin, because the first question is where the insulin is binding. So for example, this is the insulin. I'm just showing it so that it is easy to show it. So you have two choices. Either the insulin will go and bind this, or insulin will cross the plasma membrane and will go to bind somewhere else, right? So what I'll do is I'll take the insulin and I will radioactively labeled. Okay. Now, after this, I am going to do the cell fractionation. And depending upon the cell, you can actually be able to devise the different types of mechanisms. If it is a bacterial cell, you are going to use the different types of cell fractionation scheme. If it is a yeast cell, you're going to use some other tools. I think you remember that we have discussed all this when we were talking about the cell disruption methods.
Yes, sir.
And if it is a mammalian cell, you can do, right? So when you do the cell fractionation, you are going to get two fragments, two fractions, right? When one is pellet, or I will say, okay, and the other is supernet. Every time you are going to see this, okay? So after the cell fractionation, you are going to see a pellet, supernet, okay? Now, what I'm going to do is, and this will continue, right? If I do further fractionation, again, this supernatant will give me pellet, supernet, right? And I can go up to this to such an extent that it will actually going to give me a single protein. So it's not going to happen in protein 1, for example. Okay, so it's not going to happen in single shot, probably every time. Okay, and now what we're going to do is I am going to see under radioactivity. I will check the radioactivity. So I'm not going to check because I don't know where it is binding and all that, right? So I am not going to do this. I am going to just check the radioactivity and following the things under the radioactivity. For example, in the first step itself that I found that the radioactivity is here. Okay, it's not here. So I will discard all that. And then I'm going to further fractionate this. And I'll be keep fractionating until I'll reach to a point where I'm going to have the single protein, which is actually giving me the radioactivity. Okay, this means this is the protein which actually contains insulin. Now, once I have this protein and which actually contains the insulin, which means radioactivity is coming from insulin node, right? So this is actually going to give me the insulin, right? Now what I'm going to do is I am going to identify this protein I will identify this protein by mass spectrometry, right? Or 2D gel electrophoresis. Okay. The other alternative is to get the same question is if suppose you have the insulin and you have the antibodies. Okay. Suppose I have the antibodies for this particular thing, right? Then what I'm going to do is I am going to incubate these cells with insulin, and then what I'm going to do is I'm going to prepare a cell lysate. So I can prepare the cell lysate in two fractions. One, I am going to prepare the pellet fraction, and the other, I am going to prepare the supernatant fraction. Now, I can actually be able to analyze these two fractions in the presence and absence of antibody. Okay. And remember that this approach only will work if the interaction of the insulin or interaction of your biomolecule to the receptor is covalent. Means it is not going to broken down if you do this analysis. Okay. So if I do this and I run the SDS page, I'm going to get the bands. Okay. Now, if I add the antibody to this lysate, if I add the antibody, so this is without antibody, and this is with antibody. And remember that I told you the condition. Condition is that this interaction should be covalent in nature, so it should not get broken down. Now, what you are going to see is that you are going to see the bands, right? And some bands are actually going to move. For example, this band. So this band was present here, but it got shifted. Okay, it got shifted because the antibody is now binding to this insulin. Okay, and insulin is binding to this receptor, right? So since the antibody is having a molecular weight of 450 kilodalton, it will go and bind, right? And that's how there will be a shift in the SDS page. So it's actually going to show you a shift. That means this is probably be the protein of interest where the insulin is binding. Okay. And you can extract this protein, and then you can actually be able to identify this protein with the help of the mass spectrometer. Once you have done that and you understand that, okay, this is the receptor, okay, then you are actually got the first clue that, okay, this is the receptor. If it is a new receptor, for example, if it is a old receptor like insulin receptor, then anyway you know what is the downstream signaling, okay? But if it is a new receptor, right, then what you are going to do is you are going to do a phosphoproteomics, because most of these are actually going to have the phosphorylation relay signal, right? So then you are going to do a phosphoproteomics. And phosphoproteomics, while you are doing the phosphoproteomics, you are actually going to generate the knockouts, because you are going to have the knockouts. So one or other receptor, you are one or other scaffolding protein you are actually going to remove. And as a result, what will happen is that after some time, so for example, when you do the phosphoproteomics first time, so you are going to have like plus insulin and minus. So here you are going to have like these bands. Here you are going to have some bands which are more like, so these are phosphorylated bands. Right? Apart from that, you are going to have the normal bands also. So these are the proteins which are getting phosphorylated when you are adding the insulin to the, sorry, this is negative, this is positive, okay? So these are the proteins which are actually getting phosphorylated when you are adding the insulin, right? Now, when you do the knockouts of the signaling partners, right? And you will do like detailed analysis, one of the receptor or one of the protein may actually disappear, right? That means this protein, or suppose this protein got disappear, right? This means this protein is downstream to this particular receptor, this particular factor. Rest all are upstream to this. So for example, if I have now removed the X factor and I lost this protein, right? This means this is downstream to this, and rest all are upstream. That's why even if you done the knockout, the appearance of other phosphorylated protein are not being changed, but the appearance of this band is being changed. And if you do a detailed analysis like this, you will be able to deduce the complete signal transduction pathway. I know this is a little more complicated, and it's still time is also very limited, so we cannot actually be able to explain you in detail. But this is what the people do actually. So they actually generate the random mutants actually, okay? Or sometimes you actually will have the biological mutant for which it is known that under these people there will be phosphorylation happening, but it is stopping at this place. Okay, so those things actually come together, and then you can be able to answer, or you can be able to deduce the signal transduction pathway of any protein or any ligand.
Thank you. Thank you, sir.
Okay, okay. So I think we don't have any more questions, and it's good to interact with with Ishan and other students. And I wish that you all will do good in your Enzyme exam. So with this, I would like to conclude today's live session. And if you have any more queries, discussion forum will remain active until the exam. And even afterwards also, my email ID, everybody knows my email ID. So you can actually post your question through my email ID as well. And I'll be happy to answer. So with this, I wish you all for your enzyme exam, and thank you.
Thank you, sir. Bye, sir.
Bye.